Hello, everyone. Either good morning or good afternoon or good evening, wherever you are around the world. So we are currently live broadcasting from Zurich. My name is Obien from MaxWell Biosystems, and we are joined today by our two guest speakers from the West Coast in California in the US. So we have Mircea and Kate. Hello. Yes. So I'll introduce them more later on, but they will be here together with us in this webinar. And they are morning time there in California, and we are afternoon time here in Zurich. So this is what's going to happen today. First, I'm going to introduce our company, MaxWell Biosystems, and then we'll proceed with an intro on microelectrode arrays, and then move on to the next topics. So first, this is about MaxWell Biosystems. We are innovating the next generation electrophysiology platforms and solutions. And our core technology, which is these high resolution microelectrode arrays, has been developed for more than 20 years at ETH Zurich, and we have good publication track record. So we have in-house expertise here at MaxWell on hardware, software, and applications. And this is also something that our guest speakers are very powerful also in their own labs. So they're experts in hardware, software, and also in applications. For today, I think the most focus on applications will be on organoids. For us here at MaxWell, we are based in Zurich. We have our own headquarters, offices, and labs, and our users around the world are from academic labs, biotech, pharma, and contract research organizations. So every year, we also have our Neuronal Network Summit, which we would like to invite you all in. And what this is about is it's a conference, basically, wherein people around the world come to join us to be able to discuss new discoveries, good science, everything about neuronal networks. So we have oral presentations, posters, and also panel discussions, as well as, of course, social events surrounding this. So please save the date for next year, April 8th to 10th, 2024. Make sure that you have this in your calendars and you can join us. If you have any questions about it, please let us know later. Okay, so this is the outline of the presentation for today. I will introduce about microelectrode arrays, and then the presentation will proceed on modules for multimodal electrophysiology experiments focused on organoids, and then after that we'll have our Q&A session. Good. Okay, let's proceed. So here at MaxWell Biosystems, we are developing the next generation electrophysiology platforms for characterizing the function of neuronal cells, and also, of course, other electrogenic cells. And for us, every cell has a story to tell. This is our motto. And for characterizing cells, it has been very well established that imaging is one way to be able to understand how cells grow, what are the different cell types, how they connect with each other, are they maturing. This is very, very important for cell characterization. On the other side, patch clamp is the gold standard for electrophysiology, meaning that you're able to get really high quality signals from individual neurons. So we see our technology somewhere in the middle. These are high density microelectrode arrays, wherein we are able to get information from every neuron, their activity, as well as also extract morphology of that specific neuron for just by following the propagation of the action potential along the axons. So here, what you can see is an action potential initiating from this one neuron and then propagating along the axonal arbors. And these are the traces that you can get for individual electrodes using the system. So there has already been several low-density MEAs out, also developed beforehand. So these are the classic MEAs, but they are low density, meaning you're not really able to get the activity from every cell, just a hint of it. So you cannot distinguish activity from every cell. Some you miss, for example, like this green cell. Some cells are undetectable or low in amplitude because there are these large electrodes, and sometimes that becomes grounded, these signals. And the cell's small signals cannot be detected. Sometimes you are also not able to distinguish between these yellow and blue, as what I have mentioned before. But for high-density microelectrode arrays, you have electrodes everywhere. So you can really see all of the active cells as much as possible, plus more. So here you can really identify the activity coming from the yellow, blue, green, and red cell. You have always an electrode at the right spot to be able to capture the largest signal from these cells. Plus, because we have many of these small electrodes that can capture also the small signals, So we can capture the propagation of these action potentials along the axons and also small signals themselves of the cells, especially if they are just growing or differentiating from iPSCs. So these are very important for us to make sure that we have high quality data coming from our recordings. So the key advantages for high density microelectrode arrays are as follows. We can get accurate activity maps such as these. We can get detailed information on the population network activity and also capture the subcellular conduction of these propagating activity. Then you can read out across different scales from network to cell to even subcellular level. So how does this look like in terms of products? So we have two currently. We have the single well, which is called Max1, and multi-well, which is Max2. So the Max1 is really small, fits in your hand. You'll also see it in more pictures later from the presentation. It's compatible with the incubator, and you can also connect a perfusion system to it. For the Max2, it has a built-in incubator, and it's compatible with both 6 and 24 well plates. So zooming in to the core technology, which is the sensor for every of these wells. So we have in each of these wells 26,400 electrodes within this 2x4 millimeter square area, which means that we really have super high density of 3,200 plus electrodes per millimeter square with this electrode center to center distance of 17.5. So every neuron can then be captured by several electrodes at the same time, which also allows us to follow this propagating activity. Plus, any of these electrodes can be used for electrical stimulation. So you can also play a piano with these neurons using these electrodes. What is also important, because we have these small electrodes, we have very low noise readouts that allow us to really capture these activities. So very integral to our system is also the software. This allows us to, get in contact with the hardware. So we have MxW Live as our software. It visualizes and allows us to acquire high quality data, organize our experiments, and also do some analysis and generate some reports. So this is the electrode array. And here you can see the red triangles appearing whenever there's an activity detected for that specific electrode. And you can also zoom into the activity of these cells. So here you can appreciate the activity of each individual neuron and even zoom into a single one and see this waveform from that specific cell. All right. So in terms of assays, these are what we have currently for neurons. First, we have the activity scan, where we are able to identify the location of the cells. It nicely matches a fluorescence image. This is just the functional image. We can also do network activity scans, wherein we are able to identify if the cells are interconnected, if they're matured enough, and they're producing these network bursts. And finally, we have the axon tracking assay, which allows us to automatically, in the software with the system, extract individual neurons, their morphology, and also calculate the conduction velocity. So these are the solutions that we offer here at MaxWell Biosystems, and we are super happy and excited that it has been featured and used in high impact publications so far, and plus more. And there are now more than 160 articles published. These are just some examples of it, and we hope that our speakers are also soon to publish using the system that they have. And also you, our audience, we are super excited to also have you using our system and publishing very soon if you have the results. Okay, so in conclusion, we offer high content electrophysiology through Max1 and Max2, which are platforms that allow us to characterize neurons at network, cellular, and subcellular levels. And these are the different research fields where these can be applied for. And for today, we are super, super, super lucky to have two speakers. It's quite special because we have two speakers today, both from UC Santa Cruz in the US. And they will talk about their work, developments on top of our system to be able to really, encapsulate all the different ways that you can empower electrophysiology to be able to really understand the function of cells, both single and also in the network. And I'd like to introduce each of them. So first, Professor Mircea Teodorescu. So he's the Associate Professor in Electrical and Computer Engineering in UCSC, and his lab focuses on research in mechanical, electrical, and computer engineering with applications in biomolecular engineering, biomechanics, and assistive technology. So his lab interfaces biology and engineering through the use of mathematical modeling, rapid manufacture, embedded electronic systems, and sensing. So you will see the results of his lab very obviously in the upcoming presentation. And our second guest is Kate Voitiuk. So she is a PhD candidate in bioinformatics, also from the same university. And for her PhD, she's very curious about how the brain processes information from the bottom up. And Kate engineers hardware, software, microfluidic devices, also does experiments with neurons in 3D stem cell derived organoids and connectoid models. So she's very honored to be supported as an NIH T32 trainee and also ARCS Foundation Adjunct Technology Scholar. So please welcome everyone, our speakers for today, Mircea and Kate. So hello.
Hello.
I'm going to stop sharing my screen and then you can share yours.
Yes. Hi. Hopefully everybody can hear us.
Yes, very well.
So thank you so much for coming. Thank you so much for listening to us. And what are we going to do next? For a couple of minutes or so, I'm going to introduce the group, and I'm going to introduce in a very broad way what we are doing. And after this, the floor is Kate's, and she's going to explain her research and everything else. So first of all, we are very grateful because we had we learned a lot, and we love the system, and obviously we managed to do sort of very interesting things. As Marie hinted, we really hope to publish a few papers in the very near future, which is the result of working very hard for the first part of the year. Now, it's a larger group, and let's start like this. First of all, as Marie hinted, so we call ourselves brain engineer, and here it is like brain engineer and genes as well somehow. And we have a little logo in the upper right corner, which actually Kate designed at some point a few years ago. So first of all, on the left side, you see the people at the Santa Cruz, and on the right side, our very close colleagues in different parts in California, or in the case of Keith, he's not in California, he's in San Luis. But it is still, we are on pretty much a speed dial, and we try to work all the time together. We range over several departments from BME, which stands for Biomolecular Engineering. EC stands for Electrical and Computer Engineering. David Hauser, it's basically computer science at the heart. And Sophie Salama, it's molecular and cellular development biology, but she's mainly focused on the human type of organoids, and some of the things that we are showing today are the biology is developed in her lab, while Mo joined us about a year and a half as a research scientist with the Genomic Institute. And his focus right now is mainly on mouse and chicken organoids, which first we thought maybe are not as interesting as a real human mind. However, because they are maturing much faster, because everything is so much quicker, they are fantastic prototyping system. Sri Kurnia, she's in a computational media department, and she is helping us with, it's a buzzword these days, gamification, but we do have several papers on games and the education part, because kids learn better if they play. Tal, you probably saw in Marmari's presentation a second ago. He used to be at UC Santa Barbara in Ken Kosick's lab, now join us as a faculty at UC Santa Cruz. We're very happy to have him around. So this is somehow the PI team. We have lots of researchers that show up in a minute. I'm so sorry I didn't put them at the beginning. We should change that for next time. But first of all, depend where you are in the world. You might have heard of Santa Cruz. But again, I always start this way because we are close to Berkeley, we are close to California, we are in California, but we are probably not as well known as other people, except for our surfing and for our bicycle, which are really awesome. The campus is really pretty. If you ever come to the West Coast, give us a shout, and we're gonna show you everything. By the way, this surfing pier, it's very close to us. But the campus is kind of in a forest. It's really nice. It's like this. There are also the bridges, and these beautiful tall redwoods, which are the pride of our coastal California, are actually really here. It turns out that some of the since the national park, which was supposed to have the tallest one. It's closed due to some flooding. The best place in California to see the tall redwoods is our campus, the northern part of the campus. This is not the tallest tree, but this is just behind the lab. And here it is in a small picture. You see actually Kate in blue, and Jess, she's a postdoc in the lab, and Keith, it's our colleague from San Luis, was visiting. Sophie is my colleague from biomolecular engineering. And so we are just walking, mystery student. We are walking around the woods. Our exact lab, it's not, it's down 10 minutes down the hill by the sea, which again is not a bad place to be. And as a proof, we have these trees in both pictures. But now going to something more important for today than our lab, is how do we generate these organoids, and why are we doing that? And Marie mentioned there are several reasons actually. First of all, like everybody else, we try to understand how. So the goal of our lab basically is to understand a little bit more how the brain forms, and how the interconnection, how the entire neural network is forming, and everything to do with it. And to do that, we need to study somehow the biology. Obviously, in some ways, the easiest way would be to have a 2D culture of neurons, which are, for example, dissociated from some tissue, or they are grown that way. The problem with that is that the connections are not formed in exactly the same way. It's kind of flat. And okay, another version would be to have a real brain, and you are doing brain slices. We did try that as well. The problem over there is that you are kind of slicing quite a number of neurons. And again, it's unrealistic from that point of view. Then we come with the organoid, which in some ways might be a more realistic model, because it's at least three-dimensional. At least the neurons grew together. They were not forced in that position. But of course, it's not exactly a human brain or a mouse brain or anything like this. And as a proof, you see a cross-section which is stained in the lower part of the slide, where indeed you are seeing all sorts of rosettes, we call them, which are, so we have all sorts of neurons which are very much relevant for brain culture. They might not be connecting exactly the same way, but it's still, we believe, at least, other people might not, but we believe that it's a better model than a 2D model. Now, how do you understand what's happening? And how do you understand a neural network? So that's where Kate is going to explain. We tried all sorts of probes. We tried poking and slicing and doing all sorts of things with the organoids, trying to interrogate in a way or another these neurons. Now we are settled to microelectrode arrays like the one that we are going to talk in a minute. And a question that we had in the seminar earlier this morning, yes, but you have a larger ball and you are just measuring on the lower side. Indeed, that is true. The only answer to that is that the organoids are all, the neurons are interconnected. And probably Kate is going to explain more when what that matters, because even if we are trying to excite the upper part, considering this would be a cross section, where the upper part we can measure things on the lower side. There, everybody talks to everybody in that small congregation of neurons. How do we grow them? So the traditional way, you have a 24-well plate, and everybody eventually has one of these organoids somewhere in the middle. If not one, there are probably more, but they are sitting in this way. And so we are proposing something slightly different to begin with, and I'm going to explain how we go about it. So right now, the entire organoid industry is kind of a cottage industry. We have many of them. Each one is different. Each one, and a good researcher will simply select some of these organoids to go further, not everybody. What we are hoping to democratize even that somehow. So right now, you have this schematic of a 24-well plate, which is being fed using a pipette, and this is a traditional way, very flexible, very cheap. You are just somehow cheap, considering that you are not taking consideration the price of the researcher, but it's a minimal research setup required. The cons are labor-intensive, statistically weak, very few replicants, and prone to human error. Probably everybody had some story of a calamity in the lab. We have many. where something, just a researcher makes some bottles and did something wrong. It happens everywhere, so we don't want that. And then we do want to have loads and loads of rare replicants. How do we do it? Well, build a machine, because I'm an engineer, so that's why I'm collaborating with biologists, and I do see machines as useful. A cross-section of a very simplified machine where you have the organoid, for some reason green this time, just for picture purposes, sitting on media which is being exchanged using a system of pipes. And there is a microscope looking at that, and there is a microelectrode array that it's sitting on. And then, of course, what not part of this presentation, because you have the media which is being always recirculated or comes back to a bottle, and you put fresh one depending on which conditions you are, you can always non-invasively have some insight of what's happening inside the well by analyzing the media itself. Now, slightly from a different point, because we are going to talk a lot about cloud and IoT and things like this in our case. We have kind of three types of applications we're after. So there are some microscopes that we are building, and this is kind of an old one. We have loads of interesting things these days if you want to meet. We have an electrophysiology branch. This is a much older version, long before we met the people at the MaxWell. Kate was building stuff in her lab. And then there is a fluidics system. So all of these things are eventually talking to each other through the cloud in different ways. And there is a control strategy for each one of them, what to do correctly. And eventually there is an analysis level, which is usually done in the cloud. And because I keep repeating this, we are engineers, we like to have a feedback controller. So eventually all of the information goes back, and you can inform the feeding, for example, based on the information you are getting from some other type of sensors. We don't have this in the presentation today. We can discuss on the side how we do it. Now but how do you get from something as simple as this to something automated? So what we did, we made our own 24 well plates, which happen to have a bunch of microfluidics going through the bottom. Each well is designed in such a way that you have a very, very gentle way of filling and very gentle way of emptying. The organ sits in the middle, and it's exactly where it has to stay. This particular design is not interfacing with the MaxWell, but I'm going to show you the one that does. Which has the same well geometry and the same everything. The goal of this was just to, how do you grow armies without touching them? And so this sits on the microscope. There's a paper correlated with this result, or several actually. And again, you are feeding fluids in and feeding fluids out. There are many configurations here. We can discuss a lot about it. But in general, it sits in an incubator. You have two little fridges. One has fresh media, one has spent media. There is a pump in the middle with all of these beautiful red connectors. And on top of the pump, there is a little screen, which is a controller for this pump. And this system, it's sitting there for a while. How do you as a user interact with it? Of course, through the internet. So panel A, it's a developer GUI. This is what someone like Kate would use in order to set up the experiment. Panel B, it's a user GUI. And now the user GUI, the reason it has a zebrafish is because we are teaching a class. We're actually teaching quite a few classes around the world. They're helping people to understand more about the biology in places in which normally they don't have the resources to see that. And that's it's quite popular, it's quite interesting. So zebrafish, it's actually a nice experiment that in some agriculture, it's very, very early on in an agriculture community in the middle of California. They actually wanted to study the effect of fertilizers on fish. And that's the model. So you can see from home what's happening. On the right, you can see on a phone, and this indeed has a bunch of organoids which are being monitored over time. How exactly this works, again, is not going to be explained here, but we are very, very happy to discuss. And from now on, I'm going to let Kate go, because now we are going to the real science from my point of view.
Thank you, Mircea. So far, we have described the 24-well system for fluidic delivery and feeding, as well as a 24-well system for imaging. Now, we want to understand how our organoid cultures are performing and developing on an electrophysiological level as well. So we start with neurons. Next. And there are many ways that we can look into the electrophysiological activity. One way is to use probes to go into the sample. Another way, as Maria was describing in the talk, is to use planar low-density MEAs. And the third way is to use higher density MEAs, such as the MaxWell system, which we decided to choose. Next slide. So this is an example of combining the previously mentioned imaging, as well as fluidics, and also electrophysiology all in one system so that we can achieve multimodal experiments and multimodal data. Next slide. So this is an example of the microscope coming from the top. This is an off-the-shelf USB microscope that you can buy. So there's nothing special about it. And it can image from the top through the lid. And in the top left, you can see an example of what the camera sees through the sample. So that large square in the middle is the MaxWell CMOS MEA sensor. Next slide. So in order to feed the culture that's sitting inside of the MEA well, we have a custom 3D printed microfluidic insert, which you can see in purple. And that's something that we designed to fit into the chip and be able to connect to a fluidic line to deliver media into the chip, as well as remove media out of the chip. And the geometry of this well is actually very similar to the purple plate for the 24-well automated system that Mircea was showing earlier, just adapted for the single well format. And this is an example of the 3D print in real life and how it looks. And you can see there's a glass rod that we were imaging through in the previous slide. And this one actually dips into the liquid so that we never have condensations. And the media is delivered into the chip using the syringe pump that we showed for the 24-well system, but now it's adapted to work with the single-well system. And many chips can be connected together to this pump to be run in parallel as replicates. And we can schedule when we feed the chips and at what time and how much fluid we deliver to them using this programmable pump. And on the bottom left is an example of a Google Calendar-like schedule that we can make to sort when we want to feed. Next slide. So in the previous picture, you saw the camera unit, the USB camera sitting over the recording unit, as in on the right side. But we can also have individual imaging stands to lug down on CMOS chips in just isolated. So that way we can image multiple replicates, not having them on the recording unit. Next. And this is just an example of what our setup looks like in the incubator. On the left bottom, you can see that one of the microscopes can actually be fluorescent. So you can not only look at a bright field image of an organoid, but also a fluorescent image of an organoid if you have a fluorescent sample. Next. So now I'll shift to the data that we've been producing using the system. So as Marie was mentioning in the slides, we use similar MaxWell assays. So for example, in the middle, you see the activity scan of an organoid, and the colored pixels are showing firing rate on different electrodes, as well as the spike amplitude, which is on the bottom. We also like to use open source custom software pipelines for analyzing the recording data that we have from the MaxWell. For example, we use KiloSort for detecting where the neurons are in the recording and being able to cluster them in time. And we can also curate the potential neural units that were picked out from kilosort using a program like Phi, which allows us to say, is this a plausible neuron detected by kilosort, or it's more likely to be noise? And then after we're done through spike sorting and curation, we can create these spike rasters, which are shown below, where we have neurons on the y-axis. They're called units because that's what the software calls them, because they're statistically inferred putative neurons or neural units. And on the x-axis, we have time. So every single line that you see, every single dash that you see is event of that neuron firing in time as detected by the software pipeline. Next. This is just an example of curating neurons, and we're really happy to walk anyone through the software pipelines that we use, and if anyone wants to use the same pipelines. Next. So this is an example of a spike raster, and on the bottom in red, we see the firing rate in red. So along the vertical column, the activity is summed for all of the neurons and is being displayed in a graph, so you can see when the whole population of neurons is firing up and down together. Next. And the really interesting thing of this spike raster visualization is that we can look and see which neurons fired together at the same time, and therefore might be revealing some properties of how the network is connected. Next. So in green, you see multiple neurons, 14, 12, and 5 firing together at the same time. And then also the purple ones also seem to be in synchrony together. And using this information, we can generate the plot that you see on the right, where we draw a physical connection from the 2D MEA spatial recording between the locations where those two neurons were detected. And the white line here represents the strength of the correlation of how they might be connected. So everything that's shown in this web-like graph here is an inferred statistical possibility of those two neurons being connected based on recording data. Next slide. So one way to study a network and the connections of a network is to be able to perturb them. And one way that we can do this is using also optogenetics. So the MaxWell recording device can stimulate electrically, spatially, but we wanted to add another modality of stimulation, which is using light. So optogenetics is a technique where we can engineer a DNA sequence into the neurons that allows the neurons to express a new kind of protein on its cell membrane. And this protein opens up in the presence of light. So when it's hit by light of a certain wavelength, for example, green or red of a certain nanometer band, then the protein pore will open up, and that will depolarize a neuron or release the ions, or essentially make it fire. So in short, using light, we can make a neuron fire if it has this protein pore sequence incorporated. So what is our workflow for using optogenetic stimulation on the CMOS electrode arrays? We start with an organoid, and we infect it with an AAV carrying the virus coding for the protein pore. The virus comes at a certain concentration, and the more concentration you add, the more cells you infect inside of the organoid. Not all of the cells in the organoid will be infected, so there will be a mixed population of light responsive and non-modified cells. And then after several days of incubation, we can now stimulate this organoid and receive a light invoked response. Next slide. This is an example of the hardware system that we built in order to accomplish delivering light to the CMOS MEA. We built this in such a way that is easy to replicate with off-the-shelf components. So every single item that you see in this diagram can be purchased online and doesn't need to be fabricated or ordered, especially. So there are no custom PCBs or no special wiring or other things that need to happen. This is what the system looks like physically. Next slide. And also, in order to deliver light to the well, we designed another 3D printed insert, which allows us to hold an optical fiber directly inside of the well and centered at a certain height above the bottom. So this is some example. You can go to the next slide. This is an example of the data that we got using the system. So you see here again the familiar spike raster with units on the y-axis and time on the x-axis. And the blue bars now represent the times at which light was turned on. And the color of this light represents how intense what intensity we used to stimulate the culture. So this graph here represents what I like to call an intensity sweep. So we're going from very low intensity light stimulations of 1 second long to much more intense stimulation with light. And in this graph, we can see that there's some spontaneous activity in the beginning before any light is administered. And then as we have more and more intense light, we see some neurons responding more and more strongly to the light. For example, unit number 11 above the orange one has more and more spiking as it's hit with the blue light. Next. This is a spatial representation of the same data. So the colored neurons that you see in the spike raster are also colored in the spatial plot using similar matching colors. And this is a zoom in on what those neurons look like and what their footprints are. So whenever a pink neuron fires, for example, or wherever the orange neuron fires or the blue neuron fires, the electrodes pick up this waveform on across a spatial area. So the neuron is heard by multiple electrodes, and every single electrode hears a slightly different signal coming from that neuron. And we can tell a little bit about the shape of the neuron and the morphology of the neuron and how this signal is propagating, as Marie was actually earlier showing in the axon propagation assay. So this is not quite that axon propagation assay, but a similar analysis where we want to see what the signal looks like spatially for that neuron. So we have these neurons that are firing at different times and have these footprints. And we can see that in green, some neurons are responding to the light. For example, I was earlier talking about neuron 11 above the orange one. Now it's highlighted in blue. And we can see that when there's less intense light, it fires a little bit. And then way at the right, when we have the most intense bit of stimulation of light, it's firing a lot more. Next. And this is just showing a zoom up of one neuron. And the really interesting thing about this plot is that first the neuron starts responding to the light almost on the onset of the stimulation. And then as it's stimulated with a certain light frequency, it starts to be a little bit more and more late responding. So it almost seems like the neuron is getting tired from multiple repetitive stimulation cycles. Next. So now that we talked about the different modalities of the data that we can have all combined together, including imaging, electrophysiology, and microfluidics, we're now looking into connecting them together using IoT in a similar way that Mircea was describing earlier, and be able to use all of these devices remotely through a user interface, and also have automated data analysis and alerts to researchers for when there's something exciting happening. Next. And this is an example of a website that we're building in order to allow us to have remote lab experiments, remote collaborations, as well as education. Thank you. And this is our team.
Thanks a lot, Kate. Thank you so much, Mircea. Really, really wonderful data and great talk from both of you. All right, so now we're ready for the questions from our audience. So once again, please make use of our Q&A button. Write in your questions there. And while we are waiting for the questions to come, I will now open up some polls for you to answer. This will also help us understand what is your current application and also what is like how to answer your questions in terms of your expertise in using the like microelectrode arrays. So this is the first question. So we're super happy to see what is our audience mostly working on. Is it 2D cell culture, 3D cell culture like Kate and Mircea? Is it brain slices? Is it more in vivo or any other samples that you may have? Oh wow, it's almost 50-50. Okay. Oh, more 3D. Cool. All right. Thank you. Thank you for participating in our poll. And now we would like to ask if you have already used or already using a microelectrode arrays. All right. Don't forget to add your questions in our Q&A panel. Good. Okay, thank you so much for answering our polls. All right, so now let's go to our first question. So hello, Frank. Thank you so much for your questions. So for both Mircea and Kate, you can feel free to decide who answers the question. So here it is. Are you planning on going to include options to measure inside the organoids?
That's an awesome question. I have a little bit of an answer. I don't know if Mircea also wants to take it. Okay, so we've been considering that perhaps we can combine silicon probes coming from the top with the MEA coming from the bottom, which might be interesting, and maybe we can stimulate from the bottom, measure from the top, or measure from the top and stimulate from the bottom. So we've been considering that application, but we haven't done it yet. And that would require having maybe a micromanipulator that you position above the CMOS sensor and then going down with the probe. And I don't know, typically we like to do long-term in-incubator experiments. So I feel like it's possible to maybe have this micromanipulator once you set it up sitting inside of the incubator. But I think one of the reasons why we haven't considered it yet is because we would probably want to start off with a benchtop experiment. And I think we're more in terms of we like long-term experiments more.
Yeah. Thanks a lot, Kate. I think with the setup that you have built so far, there's a lot of things that you can still expand to. So really great questions. Thank you so much, Frank. Next question from an anonymous attendee. Thank you, anonymous. So she said, hi, Mircea, hi, Kate. Thanks a lot for the great presentation. I'm just wondering, how much does it cost for the whole set to allow you to acquire such results, excluding the MaxWell's MEAs? So I guess you will have to ask us about the MEAs, but then they're asking you for everything else.
I actually calculated the bill of materials for the optogenetics, and it's a little bit under $1,000, or roughly around $1,000. That includes, I think the most expensive things are the LED and the LED driver from Thorlabs that we used, and the fiber optic cable. And I think we went for a longer cable, which is more expensive, but there's also a shorter cable, which is a few $100s less. So I think it's somewhere around $1,000 for optogenetics.
Yes, and I guess you are publishing these somewhere later, right? So I guess
actually it's in review, so hopefully soon enough this is gonna be so optogenetics is gonna hopefully come through very soon. The other part that we showed you, we are gonna send it hopefully next week, Kate. I don't know.
Okay, some internal discussions here now. Thank you so much. So next question from another anonymous attendee. So I think this is specific to one of your plots, Kate. So what is the y-axis, the spike plot denote?
This is the spike raster where we have the dots over time. Is that correct? The lines?
It is.
Anonymous attendee, maybe if you can elaborate a bit more of your question in the comments section in the chat, that could be helpful.
This slide.
So we have several y-axis spike plot. For example, there's
This is gonna help more.
There is this one.
So the time it's on x-axis. The black one are the number of units, which are basically the neurons. Neuron is going to fire. This is just taking a note when a neuron is firing. We are not showing the entire pipeline, which is generating these dots. But based on a lot of magic, we find out that neuron number 14 fires here and doesn't fire for a while, fires again, and eventually fire in the same time as 12. Yeah, they're very similar. And the red one is a summation of everything. Go on, Kate, sorry.
Mircea, could you also skip to slide 47?
I think so.
Yes. Slide, yes.
So we have two different plots. One of them is the raster plot where it's showing when neurons fire. And then we also have the spatial plot to complement it, which shows what is the 2D layout of the neurons firing. So each plot has different applications or different visualization purposes. And the one that's on the bottom with the spike raster, that one is just explaining when, and it's not showing the waveform. It's just at what point in time did it fire.
Yes.
Is that helpful?
I think so. Yes, you have explained most of the plots. Thank you. If you think we have not answered your question, please add another question to the Q&A. Thank you. Now we have another one from Daniela. Hello, Daniela. Thank you for your question. So he said to both of you, hi. So he's interested in the setup to obtain images of the organoids on the HDMA surface. So do you uncover them to take a picture from the inside of the incubator, or how do you maintain the sterility of the whole thing? Yes.
That's an awesome question. Mircea, could you skip to slide? There was a slide in the other presentation.
In the other, yeah, this is what we did last week. This one.
Yes. So this is an example of the image that we get on the using the USB microscope. Sometimes we if we don't have a microfluidic attachment, a microfluidic insert on the MEA, we like to use the membrane lids because they have less evaporation than traditional lids. And so through the membrane, you might get a slightly cloudy image like this one. The imaging technique that we use for the fluidic chip is that we have a lid with this glass rod that is being submerged into the media. So through this glass rod, we're able to get a much more crisp image and also not have condensation or any clouding that you may have seen in the other picture where it's two organoids and they're a little bit cloudy. So I would say the USB microscope is nice for us, for our purposes, because we like to take a picture and then align that picture to the recording and see where the organoid is spatially. So we're not really looking at specific neurons or specific features that are very close up. We just want to get a general picture, but it's still possible to take the MEA and bring it to a very high resolution, nice microscope, and probably a high resolution fluorescent microscope would probably give the best results on the MEA.
Yeah, we didn't put any of those pictures here. I don't have any. Yeah, it's not the best microscope, but it is
an USB one, so useful. It's useful for.
It's there forever. You can. Okay.
Yes, thank you so much for your question, Daniela. So we move on to our next question. Hello, Roy. Nice to see you. So you showed around 20 identified neurons, spike sorted neurons. Were you able to validate it with some imaging?
Mircea, do you want to? I can answer, or do you want to?
No, you go.
Okay. Sure. So because we have a three-dimensional organoid and we're measuring the electrode activity on the bottom, we usually don't expect to see the neurons that we're recording from. However, sometimes the organoids like to sit down on the MEA and spread out a little bit, and sometimes they make an axonal projection. And this axonal projection could be visible on the microscope from just the USB microscope. You can see a little bit of tissue or a little bit of projection coming out, and then we can see that axon, for example, is generating some spike event. So yes and no. Sometimes it's on the bottom and we can't see it. Sometimes if it's going out, we can detect a little bit of that tissue and be able to see it.
Thanks a lot. Yes, Roy, you raised your hand. Yes. Hello. Would you like to talk?
Hi.
Hi, Roy.
Yes, I have many follow-up questions actually.
Go ahead.
So one of them is, yes, we recorded some organoids as well, and we saw this really nice halo around them while recording. Did you see that as well so that was the axonal projections that you were referring to?
I think so. How long were your organoids on the MEA?
We plated them around day 90, and they were recorded for like at least 3 months. But like after 4 weeks, that halo started to disappear.
Oh, that's interesting. Okay, so definitely if you're measuring for so long, I think there's enough time for the projections to form.
So my follow-up question is, did you ever try to put two organoids together on the chip and see if they are able to make connections between themselves?
Yes. Sometimes if you put two organoids together, they can spontaneously make connections. We can also confine them with microfluidic mold that can be put onto the CMOS array, and then you can see the projections more guided towards each other. But even without the microfluidic confinement, they can still find each other. And actually, the interesting part is that over time you can see the two sides of the MEA where the two organoids are, they become more synchronized. So, for example, if you have that raster plot where you're looking at the times of neuron firing events and you divide it by the right side and the left side, you can see that there's synchronization between both of the sides.
Oh, that's very cool. I would love to see how those rasters look. So sorry, I would have like just last follow-up question. Do you feel those 20 neurons are more or less than what you actually think are on the surface, or like are actual somata that are on the MEA?
I think it all depends on which example we take. So in this particular example, we thought it's very easy to see everything. We have some which have loads and loads of neurons on the bottom, but them it's kind of impossible to see. So we are using this as an example for the thing. But go on, Kate. Sorry, I took your.
I'm just remembering recent recordings, and I think typically if we have activity, we see at minimum 12 neural units from an organoid, which is maybe 1.5 millimeters or 1 millimeter. So maybe 12 units and maximum 60. I think that's our range from an organoid.
Okay, thank you very much. Very
welcome.
cool. Very cool stuff. Thank you very much.
So one more thing, they're changing, because I'm not sure if you notice the same thing, but they are slightly moving from here and there from time to time. So you have to recalibrate the whole thing. So some of the units are going to the next.
Sometimes for the same sample, sometimes there are more neurons and sometimes there are less neurons that the algorithm finds. And I think because things are moving around and shifting in the organoid, there might be those changes.
Thanks a lot. And Roy, thanks for the questions. I think the halo effect could also happen if you have a larger organoid as well. I think you have, in this example, smaller organoids. So maybe we can discuss more in detail later. Okay, we have more questions to come. Let's try to answer them all. So hello, anonymous attendee. So the question is, for how long can you record and stimulate and keep the neurons on the MEA with your organoids so far? Yes.
That's a great question. I think we just heard that we can keep them for months from another attendee.
Yes.
And that's also true for us. We've been keeping them for maybe 2 months on the MEA, or typically it's a few weeks.
Okay, cool. Thank you so much. Hello again, Frank. Yes, so Frank asked, is there a reason for rather long stimulation with light? 1 second is much longer than electrical stimulation. And is it possible to stimulate much shorter, say in a few milliseconds range? And he says, thank you very much for the presentations. Interesting presentations.
Thank you. Yes, it's definitely possible to stimulate for a shorter period of time. I think in the assay that I was showing, I was going for looking at intensity, and I wasn't sure how long I should deliver the light for the intensity to have an effect. So I was a little bit conservative, and I decided to choose a second to make sure that it really stimulates something. But there's also another assay that I like to use, which is duration sweep, and you can start from a long you have a fixed intensity of light, and you start from a long exposure, so maybe 1 second, and then you can go shorter, and see if you can still evoke the spike on a neuron that was active at a longer duration.
Cool. Thank you so much.
Some way, the elephant in the room, to address a question that everybody asked, and someone I did ask last time, and most likely someone in mind right now. This particular design, it's very cheap, but it is not addressing a neuron at a time. So you are flooding the entire enclosure with the same. Yeah, so you're just shining to everybody. Is it a good idea or is it a bad idea? It's a great idea in some examples. So yeah, we can talk more if someone wants to know what were thoughts about that.
Thanks so much, Mircea. Okay, so we have 3 more questions remaining. So hello, Leonard. Have you ever had problems with neurons detaching from the substrates? If yes, did the switch to automatic cell culture kind of microfluidics alleviate that problem?
That's an amazing question. Yes, we've had problems with attachment, and I think there are many ways that we've combated the problem. More recently, we actually had really good success with attachment. And I think it might be partly, I think the success might be because we use smaller organoids. And previously, we maybe use organoids that are 2 millimeters in diameter. And I think those are more likely to detach than 1 millimeter organoids, just because maybe there's more shear force, and maybe, yeah. So I think smaller organoids have higher success. I think we also, maybe it's partially just learning to handle. Yes. Yeah, so it's maybe over time in our lab we learn to walk differently, or to I don't know. So it might be, I think technician handling is also a part of it. But I think finding a protocol that works in terms of the kind of molecules you use for adhesion, the duration at which you incubate before adding media, and then how gently media is added. Actually, it used to be that I would add media at an angle because somehow I thought that was more gentle, because maybe it rolls. But then actually in the MaxWell Biosystems conference last, I think in May, I learned that it's, and then I realized that it makes a lot of sense. It's actually, if you have your pipette at 90-degree angle perpendicular to the well, and you gently pipette, that will actually create less horizontal shear force. So that is also helpful. So just a technique, protocol, and organoid size, I think helps. And with the fluidic and the fluidic insert, the way that we designed the insert, it has it doesn't deliver media to the very bottom of the well. And it doesn't take a media from the bottom of the well when it aspirates. It's somewhere 50% along the well. So we deliver media from the so we pre-fill the well with a pipette first. So we put in the 3D print insert, then pre-fill the well gently with media with a pipette, and then the liquid comes from the middle, goes into the chamber where the organoid is sitting on the bottom, and then is aspirated from the top. But because the inlet and the outlet in the well are at a certain height above the bottom, we're never pulling on the organoid that's sitting on the bottom. So we've never had attachment happen with the fluidic inserted. I think the most attachment happens when we're handling the chip or when we're doing initial steps. But once we let it be, it's been good.
We did a bit of fluid mechanics to make sure that this is actually quite a shape we want, and it seems like it is. It works.
Thanks a lot, Kate and Mircea, for sharing. So yes, so if you would like to discuss more about your protocol, whether that's for organoids or cell cultures, we are happy to discuss with you here at MaxWell Biosystems. Attend also our summit to learn more about what other people are doing and also from us. And you can always download the protocols available online. Thank you, Leonard. So we have one from Julian. Hello, Julian. So he was wondering, to what extent does the perfusion affect neural activity if you see any changes? And do you perfuse continuously or like from time to time? What is your current flow rate? Thank you.
That's an amazing question, and I'm super excited about this. So recently, we've been taking regular recordings, and we've also had scheduled feeding. And we've been looking at how the activity changes. So actually, I think slide 20.
Like this is not good.
We have slide 22. Slide 22 in this presentation.
In this presentation.
Yes. Yeah. So we had our feeding events plotted as blue lines. And then the orange is the number of neuronal units that we had during that recording. So to answer maybe also an earlier question, we do have fluctuations in the sample in terms of the neuronal units that are active and are detected by the software for spike sorting. And we see fluctuations in the number of units throughout time. We haven't yet really correlated that well with feeding. So we're not too sure, but that's something that we're exploring, and we're really excited. And when we do manual feeding, we usually find that right after feeding, our organoids tend to be a little bit more quiet. So we were expecting to see something similar here, but we're not too sure yet about the data. So it's still a discussion, and we're not we perfuse in this automated feeding system. We were perfusing every 6 hours, and the perfusion system was adding removing half the volume and adding half the volume. So similar to traditional cell culture, we're not changing the whole media, we're changing a part of the media, and then the fresh mixes with the previous. Does that answer the question?
I think so. Yes. So I guess the other question, which maybe we can directly answer to Julian later, how do you make sure that the flow is good, slow enough that it doesn't go to the bottom? But maybe we don't have much time, so let's answer the next.
The answer is very simple for the fluid mechanics. So we had that little fluid mechanics slide in which we are simulating how the. Sorry, did someone say something?
No, go ahead.
Yeah, so we try to simulate first in such a way that we are not we are not moving the organoids around. We have a simulation.
Thank you. And then our last question for this session is from Daniele. So he has another question. What do you use for sorting the firing rate, spike amplitude plot, and detected neurons? So he was not able to get the name of the software. So I think if you're interested, we can also send you the list of software from Mircea and Kate by email. But maybe you can just mention again now.
Yes, for spike sorting, we use KiloSort. And that is open source and available on GitHub. For curation, so looking at the output of KiloSort and seeing is this cluster of neural signals a good neuron, or is it more likely to be noise? So for example, if it's noise, it's going to be very sharp. So using for this, we use Phi, which is another open source software that's available on GitHub, and it provides a GUI and a way to look at the output of kilosort and be able to rank the quality of each neuron.
Yeah.
And then for the plots, we just use matplotlib and Python.
Thanks. And the next question, so after sorting the neurons, do you use other assays to measure the activity only of the electrodes with the neurons on them? Or maybe like your experimental paradigm after sorting, what are your next following day steps?
Just to repeat, so after sorting the neurons, do you use other MaxWell assays to measure activity from the neurons? So we actually it's flipped. First, we use the MaxWell assays because they're the first software to run and it's very fast. And then and then that's how we know, is this data good? Is this data not good? Should we even should we take the time to spike sort it? So sometimes we have an organoid and we don't sense a lot of activity. And we see that from the automated analysis for the activity scan, for example, that we have in the MaxWell GUI. And then we can say, okay, should we throw the sample away or should we keep the sample? And if the data is good, should we analyze it further?
Thanks a lot. All right. Thank you so much, Kate and Mircea. A lot of questions we have received in this session today. Thank you. So now we are finishing up our webinar today. So I'm going to share my screen for some final words. All right, so we have finished our Q&A and polls. So here at MaxWell Biosystems, we are celebrating our 7th anniversary. So to celebrate this, we now have discounts on our products. So please contact us directly to know which of these products we have these discounts, including you, Kate, and Mircea as well, if you're interested. And so thank you so much. You can meet us in these upcoming events, SFN, Nanobiotech in Montreux, ISSCR in Vienna. You can contact us at info@mxwbio.com, schedule a call with us through Calendly, or visit our website at www.mxwbio.com. So thank you so much, everyone, for joining on today's webinar. We will follow up soon to those. We have remaining questions to answer, and please wait for our next webinar upcoming again. Thank you, everyone. Have a nice day. Have a nice evening wherever you are. Thank you.
Bye. Thank you.
Goodbye.